Okay, so we're going to go ahead and get started. Good evening, and thank you guys for attending SimLink's Virtual Teen Science Cafe. My name is Askari Bumpus, and I'm currently this organization's program assistant. And it's my privilege to introduce our speakers for today, Beth Tuck and Cameron Washington. And today they will be teaching you guys about genetics and genomics.
Right. I'll go ahead and pull up the slides. It's a pleasure to be here.
Thank you. So as Ascari mentioned, we are both here tonight from the National Human Genome Research Institute, NHGRI. That's part of the U.S. federal government and part of the National Institutes of Health that's based here in Bethesda, Maryland. And we're really delighted to be here and chat with you. You can go to slide 3, Cameron. We'll skip this one for tonight. So I wanted to start off with just sharing a little bit about my college and career journey. So I am from West Virginia originally, born and raised in Parkersburg, which is a small town on the Ohio River. And there's a photo of my mom. She's a city councilor in my hometown now. And so service has always been a big part of who I am as a person and what I want to do with my life and with my career. My hometown is most known, however, for not these beautiful scenes, but for some pretty serious environmental injustices that have been happening over the last several decades. So this photo is an example of one of the chemical companies that had a massive warehouse explosion at one point. And then the other thing that we're infamous for is if anybody has seen the film Dark Waters that has Mark Ruffalo in it, it came out a couple years ago. That's set in my hometown, where DuPont had been putting some pretty gnarly chemicals in the water supply for decades and decades and decades. They knew they were toxic and still, we're just letting this happen. So the cancer rates in my hometown in the Mid-Ohio Valley are quite high, much higher than other parts of the country. So that's kind of the backdrop of where I was raised and what kind of stimulated me to be who I am today. My impression of science was not all that positive, right? I thought it was like memorizing the periodic table and all these chaotic things. But so when I got to college and I went to a small school called Hanover College, it's a small liberal arts school in Southern Indiana, I was so surprised to like totally fall in love with science. I learned about genetics and evolution and all these kind of things that like totally stimulated my brain in a really powerful way. And I was forced to take that class, but I'm really glad that I did because it really changed my life. After college, I went to Washington University in St. Louis, which is a pretty fantastic school for molecular and cell biology, and is also one of the main sites that completed the Human Genome Project, which is one of the things we'll talk a little bit about in our work at NHGRI. And I studied molecular and cell biology. And so I'll break here from my journey to tell you a little bit about my research focus. So when I was in graduate school, I was studying rare neurological disorders. Those are things that affect your nerves and especially cause degeneration. So usually like you start off with a functional brain and sort of lose control of certain parts of how your brain or body works. And so in this case, my mentor was a doctor in the clinic and he was also a researcher. And so what he would do is actually see patients in the clinic with these really rare conditions. And then we would bring back samples of their skin tissue or other samples of their tissues to study in the lab. And so in this, on the left side of the diagram, you'll see what's called a pedigree or a family tree. And so you can see all the squares or the diamonds, excuse me, that are colored in. Those are people who have the condition, and all the squares that are empty are people who didn't have the condition. And so what the scientists do is kind of trace the lineage of these conditions through the family. And then what we can do in the lab is actually sequence their DNA or read out the letters of their DNA and look for changes, look for of that family who has this mutation and who doesn't, who has this condition and who doesn't. And with that mapping exercise, we can figure out what the genetic causes of these diseases are. And so over on the right, you can see some photos of what patients with this condition, which is called spinal muscular atrophy with lower extremity predominance, it's a mouthful. But basically what happens to these patients is you can see they have kind of bowed legs. Their feet are a little bit abnormally shaped and they have kind of weird, unusual muscle patterns. And what's particularly strange about these physical features of the patients is that they are missing their quadriceps muscle, or it's kind of like really, really underdeveloped, or it kind of degenerates over time. And so we were really curious about like, what is it that happens to these patients? Why do they get this illness? And what could we do about it? How could we develop new therapies or new treatments? And so my role in the lab was to study mice models of this disease. So it turns out that there was already a mouse that has a very similar mutation to the ones that we found in people. And so that way we could study this physical condition in a lab mouse. And so what you see here in the bottom picture is what's called a wild type or a regular mouse. And if you hold the mouse by the tail, normally they'll splay their feet out because they're trying to grab onto something to try to like come to safety. But our little mice, the ones with the mutation similar to our patients, their feet kind of crinkle in, and they kind of have this like tension in their feet. And so you can even see that in like the person's foot in the little one labeled M. So it looks very similar, and they have like weakness in their legs and they have a little bit difficulty walking. And so with those mice, we could study to see how does the disease progress, or if we could try to develop new medicines or new gene therapies to try to treat this condition. So that's what I did in my research. But as much as I love the lab, I love teaching more. I was really more interested in how do young people get interested and excited about STEM. And how do we make sure that the opportunities in STEM are really open to everyone and not just those who are lucky enough to be in the right place at the right time or have the parents who could steer them into those paths. So I got involved in science education, and I started working at the St. Louis Center. I taught high school biology for a little bit, and I taught at Maryville University, where I was teaching a lot of pre-health students, people who are going to be nurses and PAs. And in all of these environments, I got to learn how people learn. And that was a really informative experience for me. But my career really changed when I got a fellowship at the National Human Genome Research Institute, and it's the same one that Cameron is in today, 10 years later. So that's pretty cool. And so I got to work on science education at this like national level, thinking about how do we inform teaching and learning at a really big scale. And I also got to work down at the Smithsonian, which was pretty cool, all sorts of other really neat experiences. And all that led me to my most recent position, which was at GenSpace. It's a community biotech lab that anybody anywhere can use. You don't have to be a PhD. You could be, a homeschool family or a ballet dancer or a patent attorney or whoever. You can come into the lab and play and experiment with science. And so I got to do that for about 5 years. And then just about a year ago, I came back to the Genome Institute to run our education programs here. So it's been a journey, a lot of learning, but it's been really fun. I'll pass it to
And
Cam.
as Beth, and yeah, and as Beth mentioned, I am super, super lucky to be working with her in the education branch. I'm actually currently a genetics education and engagement fellow. But before I became a genetics education and engagement fellow, I was in South Carolina. I was born just outside of Charleston, South Carolina, in a town called Summerville, and graduated from Cane Bay High School. So public schools all the way. And it was really in AP biology that I confirmed that I loved genetics. Got exposed to it in seventh grade with Punnett squares. It came back again freshman year with traits and families and how the characteristics are passed from generation to generation. And by 11th grade, when it came back again and we learned about DNA more in depth, I said, this is it. Like, this is where I need to be. And that was also the same course where I found out what a genetic counselor was and how they blended their passion and interest in genetics with being able to work with people and work with really rare and uncommon conditions. So that led me to work really hard in school, and working really hard in school, I was actually afforded the Palmetto Fellow Scholarship as one of our top scholarships in the state of South Carolina. And that led me to Winthrop University, where they offered a full ride, essentially a debt-free education to go to Winthrop. Part of being able to go to Winthrop was because I was able to enroll in this Eagle STEM Scholars Program, which was a bridge program that really sought to support people from underrepresented backgrounds when they were majoring in biology, chemistry, mathematics, and physics at Winthrop. I didn't realize it then, but looking back, having a support system and having people with similar backgrounds and walks of life and developing that community very early on in my college career was pivotal, really, to being able to get through undergrad and get through college. Um, what's pictured here is my genetics professor. So she was actually, this was her first year ever being a professor, and I ran down the hallway and said, I love genetics. Can I please work with you? So Dr. Cole has been a really fantastic mentor in being able to work in a genetics lab and being able to just learn more about genetics and working with animal models like Beth demonstrated in her lab. The reason that Quebec is on here is because I also fell in love with French at a very young age, and so I was really able to study abroad in college, which was super exciting. One of the reasons that I put this on here is because I was the first person from Winthrop to ever go to Quebec, and I still think that I am. So I had this long history of kind of like charting and blazing trails, but I'll say that it was a really good personal development experience. I learned a lot about myself. I learned a lot about working, on teams and working with people from different parts of the world. So really good personal development experience. McNair was another bridge that allowed me to get to VCU, where I studied genetic counseling and did my genetic counselor training. This is an aerial shot of the MCV campus, which is in Richmond, Virginia, which is the capital of Virginia. And they're working with a large, complex academic medical center. I was exposed to all areas of genetics, working with kids with rare disease, adults with unexplained issues, people with family histories of heart disease, for example, women getting diagnosed with cancer for the first time. So really able to put all of the Punnett squares and the genetics into practice and actually helping people using genetic testing and access to different resources. Um, After I got my training at VCU, needed a job. So I set my sights back to go to South Carolina because I felt like giving back to my home state would really, I think, pay back to all the support that they gave me going through college and getting the scholarship. And so I set my sights on the Greenwood Genetics Center, which is a nonprofit that serves the state of South Carolina. And this is a sticky note telling my friends that I got the job because it was my dream job and I was super excited. And so I had a blast. I worked at the Genetics Center for 5 years in the clinic, met many, many incredible geneticists, genetic counselors, PAs, nurses, worked on a team, and then also got exposed to a wide range of genetic conditions. And this sign here, NF1, is a genetic condition that I didn't appreciate initially, but at the end ended up doing research and trying to advocate for more research funding and clinical care for that patient population. So that was a really meaningful experience getting to work with the patients one-on-one. But throughout this story, the one piece that I thought was missing was I think that we do a really good job of, giving back to different types of communities. But I wanted to be boots on the ground, out at the festivals, out at the fairs, just really getting people engaged. And so I set my sights on this fellowship that would allow me to get community engagement experience, learn how to talk about DNA and genetics. And so I've had a blast so far working on different projects. This is a photo of me at an outreach event, but we'll get to talk about some of the other cool things that will hopefully benefit you guys, but also benefit the broader public as well. So just a quick to show about the day-to-day, because genetic counseling is a pretty popular profession, especially for folks who are interested in genetics and genomics. And so when you Google genetic counselors, you'll get an idea that they are able to use their genetic knowledge to be able to help with people. But what does that really look like in a day-to-day? So while I was a genetic counselor, I worked 9 to 5. I did not work nights or weekends, which was great. Um, I primarily had an office, but I also saw patients in a clinic space or in an exam room. These were my tools that I used. So I used a laptop. I used a phone because I would have to call patients with genetic test results. I would have to call doctors, for example, and really talk through what the results meant and what our next steps might be. Documented in the medical record, worked directly with patients, like I mentioned. I was lucky enough to be able to train up the next generation of genetic counselors by working as a mentor, and then also bringing in folks from college who wanted to become aspiring genetic or aspire to be genetic counselors. And through all of this reading, there's a lot that we still have to learn about genetics, and so did a lot of reading throughout that experience. But now that I've transitioned into this fellowship, it looks a little bit different. So I'm still using similar tools. I'm still reading. I'm still using a laptop. I'm still mentoring and giving back. But the day-to-day looks a little bit different with digital communication. So I'm sending a lot more emails. I'm on Teams more. I'm on Zoom more. But I'm also writing for larger audiences. So the documents aren't just for patients anymore. They're for everyone, for the broader public and for students. I'm also developing relationships because science is a team sport. You can't do any of this work alone, and it does take a village. And so being able to collaborate with people who are just as excited about doing good work and giving back and making a difference is crucial to be able to move the science and genetics forward. And then since I don't have formal training in education and outreach, I'm doing a lot of learning. That is through experiences, that is through webinars, that is to talking to people, that is through reading. I love to learn. I'm a lifelong learner, and I feel like I'll be learning till the day, I'm in my 80s, then my 90s, and my hundreds. So it's just been a really incredible experience to get to learn a new skill, but also to give back in a different way. And then last but not least, working on a couple projects, wanted to develop a career quiz. So for people who are interested in genetics, that would be a customized interactive type format to see where you might fit with your skills and interests in the genetics workforce. And then also wanting to pay homage to all the contributors to genetics and genomics from African American descent and from the African diaspora. So working on a way to honor and commemorate those contributions from the folks that have done great work in our field so far. And while I've been with the fellowship for a couple months, I still have a couple more months to go to make sure all of these things get out to the public.
Thank you,
All right.
Cameron.
Thanks.
So we wanted to share a little bit about our work now. You've heard a little bit about where we've been and some of the things that Cameron's been working on, but we'll also share a little bit more about our team and the things that we do. So again, we mentioned we're from the National Human Genome. Oops, sorry.
So,
Want to give a little bit of, sorry, I didn't practice this part with you earlier. We're from the National Human Genome Research Institute, NHGRI. And NHGRI, as I mentioned, is a federal agency. It's part of the National Institutes of Health. And so that means that, and NIH as a whole is the world's largest funder of biomedical research. So this group gives out millions and millions and millions of dollars to scientists all over the country and all over the world so that they can carry out their research at universities and science institutions all over the country. Our focus is on advancing human health through genetics and genomics. So we're really interested in how you can use genetic information to improve medical care. So that's kind of the context in which we're working. And then our team in particular, so you can go to the next slide now, Cameron, is focused on education. So we know that these advances in genetics and genomics are happening at a very rapid pace. And so we want to make sure that the broadest community possible is literate in the language of genetics and genomics, capable of making decisions about their health and their family and what they want to do with that information. And so we do that by engaging with students, with teachers, with different public audiences, with healthcare providers, with underserved communities, folks who've been excluded from medical care and biomedical research in the past. And especially to make sure that we are of service to these different communities by providing access to high quality education and hands-on experiences for young people. So that's what we do. And then the other function that we play, which I think is really important, is that we spend a lot of time listening to communities, to students, to educators, and we make sure then that our leadership hears that message, hears that information of what's happening in their neighborhood, in their communities, and how can we use genetic information to improve human health. So that's the big picture of what we do. And then we wanted to tell you about a few examples of the kinds of projects that our team takes on. So the first one is DNA Day. National DNA Day is hosted every year on April 25th. It commemorates the discovery of DNA's double helix in 1953, and then the final publication of the human genome in 2003. So 50 years later, we were done with the Human Genome Project. And so this day is a big celebration to encourage people to learn more about genetics and genomics, how it connects to their lives, and to do some career exploration. So we love that kind of stuff. It's our jam. So we wanted to tell you about a couple of ways in case you're interested in getting involved with DNA Day. One is that there are events that you could either host or that you could participate in. Usually these events are hosted by.
Oh.
Okay, there we go. All good. Okay, great.
Sorry about that.
No worries. So the goal of these DNA Day programs is really to help people learn a little bit about genetics and genomics and to learn more about careers. So as an example, you can see in Charleston, South Carolina, the Charleston County School for the Arts was doing a forensics DNA activity where they were extracting DNA from cheek cells, and you could hear more about what that experience is like. So you can find events near you, or you could participate, or you can lead one yourself and get that included in our national network of events. So that's one thing you can do for DNA Day. The other thing that you can do for DNA Day, and this is a little bit new for us, so we're trying an experiment this year, is we're going to host a science comedy show on at NIH, and we'll also be broadcasting it live. So if anybody's interested, you can watch it on the webcast. And so this is a comedy show where a bunch of comedians ask questions of a scientist expert, and they crack a lot of jokes and they play some games. And again, this should be a pretty fun experience to get some learning, but in a fun way, in a kind of silly way.
And then, as Beth mentioned, we also work with groups like yours, groups that have youth and folks that are interested in science broadly, and interested in healthcare, and interested in genetics and genomics. So we've been able to partner with the DC County School District as well as the Prince George's County School District to either bring folks to us here on campus, to come to you tonight, like we're doing virtually, or out in the community at schools or other community centers to be able to bring some of the programming to you, to the public. These can include things like hands-on activities. Students can participate in doing research on a genetic or genomic related topic, for example, or help, as Beth mentioned, with putting on or assisting with DNA Day, for example, in their own communities. A lot of these things can happen in the summertime, but there are also opportunities, of course, during the school year to get involved with our group as well.
So
All right.
that's kind of a lot of what we do. We get to like for our job, we get to play and have fun with science, and that's pretty cool. So yeah, so we had an activity plan, but I'll ask Ascari if we should kind of go through it as a demo instead of as an activity. Is that yeah, okay, wonderful. And we can leave you with the links to the Google Doc. So if people want to play around with it, you're welcome to do that at a later date. So our task tonight is to introduce you to the idea of how DNA codes and encodes and decodes information. How does it work? And to kind of think about it as a novel way to explore what this could do in the future. So not just about genetic information now, but what could it be? So go to the next slide. And again, just as a kind of a reminder, what is DNA? How does it work? DNA is our genetic information. It is the thing that we inherit from our biological parents, and that if we have offspring, we pass it on down to our offspring. So it's this kind of connective thread through the generations and back into history that ties us together. And in particular, DNA is a really cool molecule because it has a couple of features about it that make it really useful. One is that it's very, very stable. And so even we've been able to find DNA from 100,000-year-old bones, and you can still read it, which is pretty incredible. The other thing is that it has this really reliable way of copying itself. So that makes it a really useful storage molecule for that kind of thread through history as you pass it along to your relatives, or even as you just build a body, right? That ability for that DNA to be very stable is really, really important for these features. So we'll go on to the next slide. And that phenomenon led scientists to be inspired by what else could it do. And so this idea about DNA as digital storage actually came about. I was doing a little bit of research for this. The idea for this came up in 1959, just a few years after DNA's structure was published. A scientist named Richard Feynman was like, what if we just put information in DNA instead of having DNA have information in it already? And again, it was very, very like hypothetical and kind of lofty back then, and we didn't have the right tools or technology to do it, but today we do. And the first time that someone actually encoded information on purpose in DNA was actually in 1988, and it was an artist who was working with some scientists. He wanted to explore this kind of theme of could we do it, and if we could, how? But this didn't really take off as a scientific technology until about 2011, 2012, which probably is when many of you all were born, our watchers. And that's when the first real major advances in DNA technology started to get to the point where the cost was achievable, was affordable to do this at a large scale. And so, for example, in 2019, they encoded the entirety of Wikipedia in DNA, and they did it in a matter of a couple of days. So that's pretty cool. That was also the first year that they ever encoded a music album in DNA. So all of these things are happening just very recently in our world. And so again, to give you kind of a sense of scale, you can see here as a comparison of how that information is stored in, for example, a hard disk on your computer or a flash drive. I don't know if any of you guys use those anymore because you got the cloud. But to compare it to an iPhone, for example, it's got more read-write speed than an iPhone, and it can store almost 1 billion times more data per, size than an iPhone. So that's pretty incredible. I mean, it's just an incredibly dense material to be able to put information into and encode it. But how does this actually work? So we'll go on to the next slide to kind of explore what's the philosophy of it. So in the first panel over on the left, basically what you have is kind of computer code, right? Computer code is written in binary, it's zeros and ones. And that code allows the computer to say, this is an image of a frog, or this is a song, or this is a code to be able to actually run your computer. All of that information is encoded in binary, so the zeros and ones. And so what scientists have to do then is convert that into another code called triplet. So it's instead of zeros and ones, it's zeros, ones, and twos. And then that triplet code they can convert into DNA. And so if it's 001, then that's an A, or if it's a 002, then that's a T. And so you're able to kind of convert these triplet codes into DNA code. And then they actually use it's called a DNA printer, which is kind of wild. It can actually print bits of DNA all in the right order. And then eventually they have to stitch it together and produce it in all these other ways, but that's basically the concept. So you can go from digital information in the form of a picture or a file, and you can convert it into DNA and actually print it. Now, what scientists do then is they put it into E. coli. And that sounds kind of weird, right? But E. coli are actually really good storage molecules too, or storage organisms, because they have a very small genome or a small bit of DNA, and you can put in extra DNA into them. And those techniques have been around for probably 30 or 40 years. And then they can make more of it. And they also take up a very small amount of space. So what comes next is you would just put that DNA into the bacteria, and then that bacteria you could just store in a freezer. So that's the next little animation there. And that will basically last forever, which is pretty cool. You can store it. This makes DNA a really cool archive material. You could also then grow it. So that's that picture of the Petri dish. You can grow the bacteria back out, and then you can re-extract that DNA and then read it using a DNA sequencer. So that last machine on the bottom right, that's a DNA sequencer. And if you read the sequence, then you can recreate the file. So that's kind of the cycle of how you store information in DNA. Now, this sounds like science fiction, but it is a true story. I'll show you a couple of examples of really cool applications of this. It's kind of a really wild one. But in this experiment, what the scientists did was they took a file that was in binary code. They encoded it into DNA. They put it into some bacteria. They grew it up. And then what they did was enclose it into these little tiny glass particles that they could put into 3D printer filament. And then they used that 3D printer filament to print a bunny that had DNA in it. And then they could re-extract the DNA to make the bunny. So it's this kind of cycle of being able to have more or less a self-replicating machine, because they could do DNA digital storage in this way. So that code was how to print the bunny that they had encoded in DNA, which is just totally crazy. So
It's
there's a little
amazing.
video here of how that works if anybody's curious to look at it later. So that's kind of the context of the things in which we're working. And it sounds like science fiction, but it's real. And again, people are really kind of talking about this as important archival material, so you can make this information last a very long time. So with that, we'll go into our activity. So we can stop sharing screen now. And let's see. So I think what I'll do is screen share the Google Doc, and then what we'll do is make sure that the team at SemLink or SEMLink will have the information for folks later. So basically what we're going to do for this activity or for this demo is do some coding and decoding with with the same idea. So we're going to do it at kind of a small scale so that way you can get the idea of it and then you could imagine what scientists could do with it at a big scale. So in this activity, the simple DNA writer, some scientists and computer coders came up with those 3-letter codes for each letter of the alphabet and for spaces and for numbers. And so each set of 3 letters, excuse me, encodes for 1 letter of the alphabet. So if you want to say something that starts with a J, you're going to start with the letters T, G, C. Let me zoom in a little bit and make this bigger. There, like that. Or if you want something that starts with the letter T, the first 3 letters of your code are going to be TAG. And so what we're going to do in this activity is pick a word that we're going to spell in DNA. So does anybody have a preference for a word that we'll spell?
Cool.
Cool. Okay, great. That'll be an easy one because we got 2 O's. All right, so we're going to spell cool. And so we're going to go back up to our table. And can anybody spot the letters for C? We've got a TCA here for C.
Mhm.
And so we're going to come down here and put it in our cool letters. Okay, next up we have O. What's our O?
TGT.
Okay. Beautiful, TGT. And so that's the next 3. And then we've got another O, so we can just do that one again. And then we've got the letter L. So here we've got an ATC. Yeah, beautiful. And so this is how we would encode the word cool in DNA. It would be this code. Yeah. And then are you ready for a little challenge there? So we're going to put a new one in well, let's do this one probably. I think that's our easy one, huh? So now we're gonna back encode. So we're gonna take this code here and we're gonna try to figure out what it says. So what I'm gonna do is go ahead and paste this into the chat and you can help me read it out and we'll figure out what those letters are. Okay.
Mhm.
So what are those first 3? TAC.
D?
TAC is a D. I'm just going to put it down here. And then we've got, oh, here, why don't I do this? And then we've got, we've got those ones done.
Mhm.
Next is CTC.
Is it N?
Yes.
Mm.
And what about our third set of letters? ACT.
A?
Yep. So that
There
one's
we go.
DNA. So, again, you get this idea of like, our codes are interchangeable. If you know one code, you can kind of apply it to another code. And that's a big important message that we have about DNA and how DNA is really a computer science field now, which is kind of cool. All right, you ready for one more challenge?
Bring it on.
Bring it on. Okay, great. So for this last challenge, we're going to go to a tool that will allow us to do this a little bit faster than our own little brains can do it, right? So we're going to go to this website that is a DNA writer. And so what we can do is either write messages in DNA. So would you all give me a message to write into DNA? You think of any
This is fun.
This is fun. Exclamation point. Create code, and then it'll give you the code of what your message is based in that DNA. So again, it's just kind of a little bit of a fun way to play around. You could write a sonnet in here. You could have ChatGPT tell you something interesting, and you can kind of think about like what does this mean? But the other fun thing that you can do is actually translate bases back to text. So the other like activity that we put in here in case there's a curious person is you could take these codes that we've developed. Why don't I pick this one actually? And you can put it into this decoder. So you can put the sequence in, and then you can ask it to translate it back out to you, and it will give you a message. So this one says, pick 5 emojis that represent your week and paste them into the Google Doc. So if you want to do that, you can kind of play around with it. So you can practice kind of encoding and decoding information. So I think that's it for our presentation. Is that right, Cameron?
I think so.
Yeah, but we'd be happy to kind of, do any other question and answers or anything else that you think would be interesting to tell the students about a career in genetics and genomics or a career in the federal government. That's also something, we can talk about what it's like to work, um, to be a public servant.
I do have a question. So you guys use like TAC, and I know the T stands for thymine.
Mhm.
Um, there
Ah,
was
yes,
there was another letter that I'm forgetting, and I think it switches when it becomes mRNA.
that's
What was
right.
that called?
Yes,
I think it's L.
Cameron.
Lysine?
Ooh, good guess. So,
Oh,
yeah,
yeah.
yeah.
Okay.
Uracil, that's right. So uracil is the letter that's only in RNA, so you don't see it in DNA. Or if you do, that means there's a sign that something's gone wrong. Yeah, usually that means like the DNA is like very decayed, or is mutated in a weird way. But lysine, as you mentioned, is actually an amino acid. So that's one of the building blocks of proteins. And so one of the things that our DNA does is have like genes encode information to be translated into proteins. Those proteins do things in our bodies that often give us our traits. So, for example, a protein called melanin is really important for hair color, skin color, eye color. It produces pigment, dark pigment in our skin, and in our hair. And so that melanin, if you have one version of the melanin gene, you have darker skin. And if you have a different version of the melanin gene, then you have lighter skin. And that's like an example of just like a change in the DNA translates to a difference in the protein. And then that difference in the protein influences your traits. So I love that you brought up lysine. That gave us a chance to talk about the genetic code. Yeah.
Um, I think that's the only questions that I have for you guys. Thank you so much for your time on behalf of SEM Link. And we hope that you guys, might want to volunteer with us again in the future. We do have different events that, we do in different formats. We also work with micro schools and things like that. So if you guys are interested, I would definitely contact Carlin Pounders.
Thank you so Yes. It's been such a pleasure to be here with you tonight. Thank you so much.
Thank you guys.
Thank you.
Enjoy the rest of your night.